part. Sorry. Okay, thank you. I'd like to thank the organizers for inviting me again to speak here. It's a real honor and pleasure to be here again and to see all of you. So today I will maybe talk about some things that are different from my previous talks, and in particular about data fusion of different types of data. So I will just briefly motivate, because motivation is not needed here, and then I will talk about new methods, how to fuse multi-omics data. And the first will be on how to fuse networks on genes, so the same type of entity. Then how to fuse heterogeneous data entities in a patient-centered way to improve various tasks of precision medicine at the same time and computational cost. We'll see several diseases, also single-cell data, etc. And then in the end, I will talk about the newest of our directions about network embeddings and how to use it to learn about function, disease, etc. So let's slowly begin. We've seen that technological advances have yielded an astounding harvest of various molecular data, from genomes and epigenomes to transcriptomes, proteomes, metabolomes, exposomes, phenomes, even now allergomes, foodomes, all of these omes. So basically, we've been amassing the data at unprecedented rates. And now, of course, the question has been for many years how to extract as much information out of each individual network type, whether it's co-expressions in particular tissue or in disease, or whether it's physical bindings, protein-protein interactions, or epistatic interactions where mutations of both of a pair of genes yield a phenotype that's not expected by mutating individual genes, metabolic interactions, all sorts of ontologies, etc., etc. So basically, the problems are computationally intractable, usually dealing with large graphs, large networks, and this is why we need to design approximate ways or heuristics. But even when we extract as much information as we can from each individual omics layer, then the question is how to fuse them all together and see what they collectively tell us, because each layer is just like a different pair of glasses that we put on to look at the same thing, at the human cell, and to learn about our biology and disease, etc. And even if you can fuse them, how to do that in a patient-centric way, basically to improve tasks of personalized or precision medicine. All right, so this is what I'll talk about today. And now let's slowly begin. And the reason why I go through this, so I'll start from one paper about 3-4 years ago now. So I'll go from simpler approaches to more and more complicated. This is why I'll intermix them. And this particular paper is from Nature Communications in 2019, where we introduced a new model of an integrated or fused cell that we call I-cell, for an integrated cell. And so basically this will be the first example of how we use that concept of an I-cell, and in particular in cancer, from this paper. And this was supported by my ongoing ERC Consolidator grant, and we have also obtained a proof of concept grant to try to commercialize these results, but that is not as easy as one might expect. Okay, so let's see what we have here. We have 3 different types of networks: gene co-expressions, protein interactions, and genetic interactions coming from these databases. So so basically, we make them tissue-specific by laying over the tissue-specific data from the Human Protein Atlas, whether it's for cancer or the origin tissue, control tissue. And then you will see how we fuse them into a new model of a network, and then we will compare cancer and control using graphlets. And I will not talk about graphlets, just I'll have maybe one slide to remind you. Now, as I said, different data types complement each other. And here in this paper, you can see that these types of networks, they overlap in around 3,000 of genes, but they have one shared interaction. And this is no problem with the noise in the data or whatnot, incompleteness. Basically, they measure different things. And this is exactly why previous methods fail, because there is no overlap. And this is why we need to make new methods. So, as I said, we will create tissue-specific networks for each of these protein, protein interactions, co-expressions, genetic interactions, tissue specificity from Human Protein Atlas. Note that this is not microarray or RNA-seq, this is low throughput. So if you don't see your favorite gene identified by our analysis, don't freak out. It's, it's only represents small number of patients. So basically the data was not there. That's why. So there's nothing wrong with the method. So we will consider only genes with expression available in the Human Protein Atlas and more than one protein-protein interaction. Okay, so nodes are genes expressed in a particular tissue, and they're linked by these 3 different kinds of interactions. And then we will create these for 4 most prevalent cancers in human: breast, prostate, lung, and colorectal, and the control tissues of origin. And this is just a slide showing you that these are large networks. These tissue specific are large, like let's say around 10,000 nodes, tens of thousands of interactions, so we can do our methodology. And this is the methodology. One slide on the methodology. So I color-coded these networks: protein interactions, co-expressions, and genetic interactions, red, blue, and green. They are represented by adjacency matrices, and then we basically decompose them into lower dimensional matrix factors, where this is genes here, and then here is something low dimensional, and then the middle ones basically are different, and they are viewed as compressed representations of these adjacency matrices. And how we do that is by minimizing this objective function. So in a standard way. And then once we get this G matrix factor, then we multiply it by G transpose, and then this is our I cell. This is the new network. And here you can see how it's functionally coherent. This I cell, this is reactant pathways. Pathways are biological process, color-coded. You see that basically the clusters just visually. And actually, additional functional information emerges from the fusion despite of no network overlap. So when we did the clustering, you can see here that in all cancers and controls, this pink one is from the I cell. They are more enriched in functions than the constituent data types, than these original networks. So this is a very nice property that we like. And here is an illustration of how much each of the data types contributes, the data sets contribute. So basically, if you only use a single data type, this is how much enriched you are. And then as you add more data, you get more enrichment. All right, oops, sorry. Now what we do here is we do further analysis. So between cancer and control, cancer can silence or activate genes, or in both cancer and control, they're not active, or in both they are expressed, perhaps at different levels. And then what we noticed is that only these always expressed genes, maybe at different levels, but I'll talk about that later, are enriched in cancer drivers. And then we investigated their involvement in cancer. How? By finding the most rewired, in each of these data types, PPI, GI, COX, and I cells. And we examined that, so basically between cancer and control, and we examined that using graphlets. And graphlets is the result of my starting grant that finished right before my ERC consolidator. They're still the best performing, robust. We've shown that these networks are geometric very early on, and now we've seen all this geometry and embeddings, etc. But we were one of the pioneers there. And basically, graphlets are used as feature vectors in many machine learning algorithms. We generalize them to directed, probabilistic. We made graplet Laplacians, generalized spectrum embedding, clustering, eigencentralities, etc., etc. Also, we went to hypergraphlets, abstract sequential complexes, etc. So this is what I will say about graphlets, and now let's go back. So we find between cancer and control most rewired in terms of graphlets, and then check enrichment in drivers. So only in I cells wiring is indicative of involvement in cancer. And this is why now we did biological validations in the lab. We took 20 of the most rewired in cancer I cells, because that's the only type. So basically only in I cells. So not in PPI network, not in GI network, not in coax, only in I cells. Rewiring is indicative of involvement in cancer. So we found the top 20 of the most rewired in cancer eye cells for the 4 different cancers. There's not 80 of them because some overlaps, so there is 63 of them. And basically almost all we validated either through literature curation or through our own knockdown experiments followed by cell viability. So for instance, in this cell line, if you knock them down, they significantly reduce cell viability in cancer, and in this cell line, significantly increase cell viability in cancer. And also we went through patient survival, and basically there are differences there as well. Note that only 17 of the 63 were differentially expressed in cancer. So even differential expression carries limited information, and this is why we need to do data fusion, and this is the power of data fusion. And then in the interest of time, I will not talk a lot about this. Basically, we went to all cancers from the Human Protein Atlas at the time, so 20 in total, these ones, and we repeated the same thing, and we got exactly the same results, and this is just an illustration of our top prediction, for which it was not known that it has anything to do with cancer. But then we went to the Human Protein Atlas, and we found significant patient stratification in terms of survival for 8 different types of cancers. All right, so we've seen a new model of a cell, eye cell. We've seen that all data types are limited, so we need to go beyond. And then I will show you now how you can utilize this in other diseases to analyze single cells, et cetera, track the dynamics, et cetera. Okay, so now the second example of this is for COVID. Basically, this is our paper that just got published earlier this year. Exactly the same model, except the tissue specificity comes from these cell lines, for controlled infected cell lines, and also from lung samples of COVID-19 positive patients and healthy individuals from the first study that got published early in the pandemic. So exactly the same thing as before, except now we want this matrix factor of genes to be orthogonal or orthonormal. And you will see why a bit later on. Again, only in the eye cells. Basically, the eye cells are more enriched than any of the constituent data types. Then these graphlet-based signatures between healthy and control, we find their rewiring. It's biggest in eye cells. And then we do the second step, and now we are going to combine another data type. So far, we had just genes and different kinds of interactions on genes. But now we are getting drugs, so drug-target interactions. So we pre-compute this eye cell, and then we put it in here, fix it here, and then we compute these. We approximately decompose these into these factors, and this is chemical similarities. This is basically the network of drugs and their chemical similarities. And basically you do this by minimizing this objective function. And in this way, we repurpose the known drugs to human genes. And basically the idea is, because antivirals are difficult to develop, but we already have very many drugs targeting our proteins, basically to bring these into the human cell. Others did that as well. Okay, now another illustration. This is a paper also published maybe 1, 2, 3 months ago in rare thrombophilia. This is a very rare mutation, so-called Belgrade mutation. It's only present there and northern Italy and in the Far East. So in rare diseases, we have very few patients, very few samples, and this can be used to boost the signal. So in this particular case, we only had 5 individuals, 2 families, 2 brothers, 1 diseased, 1 healthy. But the healthy brother had this diseased daughter, and then another family was of 2 sisters. So, there's basically no statistical power to get anything from the genomes here. And then we utilized eye cell is here. We have our networks, protein interactions, co-expressions, genetic interactions. But then we have patients and their germline mutations. So we factorize this matrix as follows. Here is your eye cell here, and then we impose this orthogonality. So we have hard clustering. P provides the cluster of patients for patient stratification, and G cluster of genes. And just an illustration of the result, that this is a cluster that's specific to all diseased people, and these mutations are specific to the healthy brothers, so possibly these are protective. but that is subject to future research. And now let's fully go into heterogeneous entities to improve precision medicine tasks. And the reason why we focus on this machine learning method, non-negative matrix refactorization, is a machine learning method, but it's explainable. It provides mechanistic explanation. This is just a generic formula that we haven't used. So basically, when you have different data types, let's say patients who have received or can receive particular therapy, then you have drugs, you can have chemical similarities on the drugs or whatnot. Then you can have drug-target interactions going here, or mutations or methylations or whatever you want between the patients and the genes. On the genes also you can have various types of networks. On the patients as well, whether it's their clinical profile similarities, etc. And then, for instance, if you want to repurpose this drug to this particular patient, the doctors will ask you why. But mechanistic explanation is obtained from here. Basically, you can say, this is because of these high weight reconstructed entities across. So basically, the patient has mutations in these two genes that the drug binds to. So not only that, this methodology captures all systems level data. They capture how data relate. Because we capture how data relate, that means that we have fewer parameters to tune than deep learning methods, which is nice, and we provide mechanistic explanation, and there is more. Okay, so this is our early paper from PSB several years ago now, where we took serous ovarian cancer patients. There were around 350 of them from TCGA at the time. We decomposed their somatic mutation profiles and drug-target interactions. As always, we imposed penalties from the molecular interaction networks and drug chemical similarities for the minimization function, basically to go towards where there is edges, where there is data. And this is just some result of drug repurposing. Oh, I forgot to say, so from the same minimization function, you read 3 tasks of precision medicine. So patient stratification from this cluster of genes, then cancer gene prediction from the cluster of genes, and drug repurposing from these reconstructed entities here, drug-target interactions. And just an illustration that this is very modular. This is, so basically conceptually how we think, and you can see which data type contributes and which one doesn't. And in this particular case, they all contributed. So this is how your area under ROC and precision recall curves are if you only use drug-target interactions to predict them. But then as you start adding more data, you get the best results when you include them all. Now, another example, so this was for cancer, now for COVID-19. Early on, we had these data, but now we had the data from the Krogan's lab of the interaction map, basically of viral proteins interacting with the human proteins, and also the differentially expressed gene from the same study as I mentioned before. And again, the same framework. We have various networks, but this basically we have viral proteins interacting with human. We have molecular interaction networks of human. We have drugs, drug-target interactions. We want to repurpose, again, drugs to the human gene to try to ease the symptoms of the disease. And these are the databases, in addition to those papers that I just mentioned that published the viral host interactome at the time. And again, so basically we simultaneously factorize viral human genes into matrix factors. We constrain with molecular interaction networks. We share the matrix factor to ensure data fusion, and these are drug-target interactions. So we have co-clustering property here for genes and drugs, and then matrix completion for drug-target interactions. But here, interestingly, it is not the human proteins that interact with the virus. It is not the differentially expressed genes in the lungs of human that are enriched in viral processes and should be treated by drugs, but it is actually their common neighbors in the interaction network. And we have checked for this, basically only the common neighbors are enriched in viral functions, and they're very different topologically in terms of average degrees, centralities, etc., also in terms of most graphlets. And then this made us zoom in more, and we had about 150 drugs and about 50 genes that we proposed for repurposing, and then a lot of them got validated. They were already in government trials in the US or Corday database, etc. And there is a nice biological story on the genes that they all converged in the nitric oxide, but I will skip this in the interest of time. And these are our top predictions. So you can look this up in this paper from a couple of years ago. Now, before going to the next section, let me just tell you about a new work of my student Katarina Mihailovic, who is here in the audience, and we also have a poster. She's a Marie Curie student on a Transis project, and this is in collaboration with Alex Skupin's lab at Luxembourg. So what they do is they take cells from patients, so basically with mutation and control. These are neurons, they differentiate them, they make induced pluripotent stem cells, and then they redifferentiate them, making the neurons. And then at the time points, they measure single cell expression data. There's other omics data that they not measure, but here is, so this is control at day 0, 6, 15, 21, and Parkinson's disease with this particular mutation, PINK1 mutation. There are other mutations, but now is this one. Again, at these same time points, how many genes and how many cells. And basically, this is an illustration of what you can do with the same methodology. You can have all of these generic, so to say, data, protein interaction, genetic co-expression, metabolic, whatever you want, but also genes to single cells. So this is the expression matrix. You decompose them simultaneously, you factorize them with matrix defactorization, and you keep this factor the same. And then basically what this results in, these are actually the embeddings. So now we are slowly changing gears into the embedding story. And you can view these vectors of genes. You can, in the embedding space, you can cluster them. So basically they come here from this matrix. But not only that, you can transform. So if I go a little bit back here, if you multiply these, you can transform from this space spanned by the genes into the space spanned by single cells. So you're going to get these gene embeddings, and then you can track the movement across the conditions. So we have a couple of papers in preparation on that right now. And this brings us to our most recent work on network embeddings to learn about biological function and involvement in cancer. And this is 2 recent papers. One is by Alexandros Zenos and others from my lab that we published 2 years ago. And the other one just appeared maybe 1 month ago by Sergio, Alexandros, Noel, and others. So I will briefly present these 2, but Sergio is in the audience. He's giving a talk, not next one, but the one after, and that will be the continuation of what I will introduce now. Okay, so what do we want to do here? We want to ease the computational burden of mining these large, complex data. And more we hear about network embedding methods. So what we want to do, we want to map our, 20,000 genes or proteins into something much smaller dimensional, d-dimensional, to use some embedding algorithm, you embed into some space, and then use those embedding vectors in downstream tasks, machine learning for clustering, link prediction, or whatever else you'd like to do. But here we wanted to do something different. We asked different questions, and everybody's now using transformers and stuff like that, which posed many questions and controversies that we have seen earlier this year. We wanted to do something else. We were inspired by a natural language processing method that was abandoned about 10 years ago, but basically at the same time when it was introduced almost. So, and it's this word2vec that is based on the skipgram neural network architecture, 1 layer. And that was observed that the words, when you push them through this, embed them in the space, the semantically similar words are close in the space based on the cosine similarity. And then there were other things that were noticed there that I'll mention just now. But then very soon after that, this was generalized because in networks, we don't have these sentences. So basically in the text, when you have the text, you have some window, you slide that window, and then you see which words co-occur in the window, and then you have some mutual information matrix that you do stuff with or whatever. So But we don't have these sentences in networks, and they were approximated usually by random walks. And then you approximate with a random walk, and then you employ the same neural network script RAM architecture, and this is the basis of deep walk, line, node-to-vec, etc., etc. But 10 years ago, in NLP, the following three things were observed. Class separability, in the sense that vector representations of elements belonging to different classes are linearly separated in the embedding space. Then, the following two are very interesting, which is vector summation. So basically, if you embed words, you get their vectors, you sum them up, you average them, and then you can in the space represent sentence or paragraph, etc. So that is very interesting. And then, but even more interesting is the word analogy. So basically that the queen is to a woman, the same as the king to a man, which means that these vectors corresponding to these words, you can do linear operation. And basically when you do this, vector of queen minus vector of man plus vector of woman is the closest to the vector of queen than any other word vector. And now we asked, does this hold for biological data, or biological network data? And can we use this to answer biological questions using simple vector operations? Because once if we embed this, if we learn this, then if we linearly can learn this, then we are in good shape. Now we have a problem of heterophilic data sets, because networks in biology tend to be heterophilic. So what is heterophilic? Homophilic is when nodes belonging to the same class cluster together, they are connected, and heterophilic otherwise. And it was shown that network embedding methods perform poorly on heterophilic data sets. And also importantly, the methods were usually tested on toy examples, while we have very large data that are noisy, incomplete. Our annotations are also noisy, incomplete, overlapping, etc. And our networks are heterophilic. So let's see if this is applicable, and if so, how. Okay. So we will investigate biological relevance of the embedding of biological networks, and also applicability of linear operations on the embedding vectors. But along the way, we initiate embedding biological networks using NMTF. Why NMTF rather than neural networks? To control the embedding space, orthogonality of the axis, and have explainable machine learning method. We have seen a lot of controversies because transformers are not explainable, etc., etc. So, and also we propose alternative matrix representations to better embed heterophilic networks based on graphlets. And we directly embed also annotations in the space spanned by genes and provide a better functional organization of the embedding spaces. So let me just illustrate things. So we want to better disentangle the embedding space. So this is why we want to put orthonormality or orthogonality on this basis, on the basis, so that the functions will be more disentangled. So any network representation here, X, we approximate like that. And now there was an interesting result from 5 years ago. Basically, it was proven that skip-gram neural networks based embeddings are implicitly factorizing a random walk-based mutual information matrix. And this is the formula, PPMI, mutual information. So there is a closed formula. Now, in this formula, there are these standard things, but focus on the adjacency matrix here. Now, for homophilic networks, random walks make sense because adjacent nodes in the network share function, but not for heterophilic nodes. And because quite some time ago we have shown that similarity in the wiring patterns can mean similarity in biological function based on graphlets. Now, instead of randomly walking along the connected paths, we randomly walk between the nodes that have similar wiring patterns. So in that formula that I've shown you, we'll replace the adjacency matrix with the graphlet degree vector similarity matrix. So graphlet PPMI. Okay, so this is basically the essence. So we will test the effects of network matrix representation, whether factorizing the adjacency matrix or PPMI matrix or graphlet PPMI matrix gives you good results. And here are just the results for Go biological process, gene ontology biological process, where you get the clusters based on cosine distance and k-means, that are the most enriched. So it says that network embedding space for human PPI networks allows for class separability. And here the best results are obtained for the standard random walk-based PPMI matrix representation. Now, the second benefit, let's test for orthogonal spaces. What does orthogonality bring you? So we embed the same PPMI matrix, but with orthogonal NMTF, normal NMTF, and deep walk, okay, neural network. And then we can see that both in terms of percentage of enriched clusters and percentage of Go terms, basically orthogonality constraint leads to more disentangled, more functionally coherent embedding spaces. And now an interesting part, the sum of vectors. You sum the vectors or average them, and we tested whether if you do that for protein complexes, whether you can predict new proteins belonging to protein complexes, and also if we do this for cancer drivers, whether we can predict new cancer, genes involved in cancer. And yes, you can. So for protein complexes, it's fine, because connectivity here is more information. So here is the normal random walk-based NMTF provides the best prediction accuracy. However, you cannot do this in cancer. So for cancer, the best results you get with this line here on the top, which is the graphlet mutual information matrix, the one that I showed you, where you randomly walk between similarly wired genes. And basically, we validated our predictions in the literature, but the take-home message is that by simple vector operations, you can do things once you smartly embed in the space. And now the second paper is by Sergio. So here is what we did so far. We decomposed orthogonal space, but now we have functions also. So we have this embedding space on genes. Genes have their functions. We have gene ontology, biological process, Go annotations of genes, and we want now basically this pre-computed orthogonal matrix basis. Basically, we copy it here and we factorize by this fixed, basically this ontology matrix of annotations, basically to embed the functions into the same space. So in the same space, genes and functions. And then what we want to do is we find these angles between the functions, and basically their cosines. We put this into this functional mapping matrix. Then we do this for control PPI net and for cancer, similar to the previous ones that you saw in the eye cell. And then basically we have these vectors of the functions. We find their cosine distances, and we define this as a moving distance. And then basically, so this is that moving distance. You can look it up in the paper. There's nothing too smart. And then basically here between cancer and background, you see that there is difference in this movement in space. We find the most moved ones, and we have a biological validations through literature curation. You can talk to Sergio more about it, but he'll present the next, the continuation of this today in a half an hour or so. So we can identify cancer-related functions now using simple cosine distances and vector additions and subtractions. So not only we identified cancer-related genes, but those functions that move the most, we also predict to be cancer-related, and we validate that they're cancer-related. Now, to summarize, so we initiate the embedding of omics networks using non-negative matrix refactorization. We show the importance of orthogonality constraint on the embedding space. We do new graphlet-based mutual information matrix that's relevant for embedding of heterophilic networks. And then we have shown importantly that this embedding space exhibits linear algebraic structure, class separability, and vector summation. And this is paving the road towards solving complex biological questions using simple linear vector operations in this pre-trained space. And there will be more coming from our lab on this later this year. And now to conclude, this NMTF is a very versatile machine learning method that has many benefits, including those that basically by carefully mapping this universe, the relationships between data entities, you have very few parameters to tune. And then also you can impose orthogonality, you can fuse data types, etc. And you can put anything you want in it. It's modular. You can measure the contribution of each data type. So you can put any DNA elements, I don't know, QTLs, you can put expressions, methylations, copy number variations, clinical profile similarities, treatments, adverse effects, whatever you want. However, each time it corresponds to a different NP-hard continuous optimization problem. You need to propose objective function, solver, prove convergence, correctness, and optimization is slow. But we have a supercomputer in-house, so we can do it. So biomedical data are complex, heterogeneous. More is coming all the time. It keeps being at the top of scientific interest. We've seen the shared vision of the World Economic Forum that was published in January this year, basically, where they have as one of the main pillars big data and medicine, and especially applied to neurodegenerative diseases that now we have begun doing in my lab. So the take-home message is that each data type is limited. It carries complementary information, and this is why they seek principal joint organization mining within the same framework. And in the end, we have to keep training embedded data scientists who speak all of these languages of mathematics, optimization, computer science, high-performance computing, medicine, biology, etc. And this is a book that we published 3-4 years ago now. And in the end, I'd like to thank for funding various organizations that funded me, especially ERC. Also my group members. Without them, none of this would have been possible. So I thank them. Many of them are here. You can talk to them. And also collaborators. And of course, we are hiring. And if you like, please talk to us. And also the learned societies that recognize this work. And thank you all for your attention.
Thank you very much for this very nice talk. I'm allowing one very short question, and all the others, please find Natasa in the coffee break.
Thank you. As always, lots of very interesting gems in this talk. I'm thinking about your random walk between the graphlets and how you define your graphic similarity. Of course, it's intuitive to have similarity between similar types of graphlets. Mm-hmm. Have you looked at maybe doing controls where you flip that to look for the results you would achieve with walking between different types of graphlets, or maybe even to try to search that graphlet jumping space to discover maybe non-intuitive similarities?
Excellent points. Thank you so much. This particular one is based on all graphlets, so like feature vectors based on all graphlets, but exactly what you're saying. Different graphlets are specific of different functions, and we would have different similarity matrices for different graphlets, and there are people in the lab working on that. There was a work published on it, like, I don't know, maybe last year, Sam Windels, who should be here also with us, and you can talk more to him, etc. But very good points. Thank you. Mm-hmm.